Hello everyone. Today we are going to study about the structure of tRNA. tRNA, also called as transfer RNA. It is called transfer RNA because this is responsible for bringing your amino acid from the cytoplasm to the ribosome for protein synthesis. So we are going to study about the structure. First of all, how is the tRNA synthesized? During translation, there are some genes which are specific for tRNA. When these genes are transcribed into an mRNA, this is a gene, so it is a DNA and it is a double-stranded DNA, of course. Then we get an RNA, okay, from the process of transcription. This RNA is cleaved into smaller RNA elements, and these give origin to your tRNA. Because this is a single stranded, the tRNA is single stranded, and you see there is no pairing or anything, this structure will be called the primary structure of tRNA. Okay, now what we are more interested into is the secondary structure of tRNA. The secondary structure, the secondary structure or 2-degree structure, okay, is resulted of some base pairing. It results from base pairing. Base pairing between what? Base pairing between this element. You will find that there are few elements here which are complementary to other elements, and what end up happening is they join together to form something like this. This part is called a helix because this is double stranded, while we see there is also a structure which is not bounded, which is very free. Okay, this is called a loop. Because this looks like a hairpin, we also call it a hairpin loop. Hairpin loop. Hairpin loop can also be called as an stem loop because the helix is also called a stem. Now let's look about the actual structure of tRNA. This is called the clover leaf structure. It is called a clover leaf because if you have seen a clover leaf, it looks somewhat like this, and this structure resembles very much to this, and that is why we call it a clover leaf structure. So because this is an mRNA, it has a 5' end, it has a 3' end. So this is the 3' end, this is the 5' end, and there are some base pairing here. Okay. Then there are loops where we see no base pairing. There is also another structure we will see, which is somewhat like this. Okay, now let's talk about it. What is this structure? Okay, so this, as I already mentioned, is a helix, while this is a loop. Okay, now every loop and helix has some function, and they have a certain name and certain modifications. Okay, so starting from this side, okay, this is called T loop. The loop closer to the 3' end is called T loop. Is called T loop. Now T loop has some modifications. What it does have is a C sigma T. Yeah. This structure that you can see, this T loop is also called as T psi C loop. Why? Because it contains what we call an unusual nucleotide, an unusual base. This is called pseudouridine. Now pseudouridine is present in between C and 5. This is from 5 dash to 3 dash. Okay, if you get to the 3 dash, we will see that C comes first, then psi, then T. Okay, what is the responsibility of this thing? It attaches the tRNA to the ribosome. Okay, another arm we can talk about is this called the D loop. The D loop is very variable. Okay, because it has a very good function that what it does, it recognize amino acyl tRNA synthase. As we know that amino acyl tRNA synthase is specific to amino acid, so every amino acid has a different amino acyl tRNA, and that is why we need a loop, D loop, which is very variable. That means you will find a lot of variations, and every variant corresponds to a different amino acyl tRNA synthase. This enzyme is responsible for charging of tRNA, which means this amino acid is put into the 3' end of tRNA, so that the tRNA is loaded with the amino acid, which can then deliver it to the ribosome, which is the fundamental reason why we have tRNA, which is the fundamental function of tRNA. Why is this called D loop? The D here stands for dihydrouridine. This is dihydrouridine is another of our unusual bases. If you love to know about the structure, it is very easy. The dihydro, as we know about the structure of uridine first. If I say that this is uridine, okay, and here we have the sugar, okay. So you can see we have a double bond. If you break this double bond, okay, so while the double bond is present, there is one H here. If you break this and add another two extra hydrogen, it would be called dihydrogen uridine. Okay, how about pseudouridine? Now pseudouridine just have a very different attachment site. Okay, in normal uridine, as we saw, the attachment site is 1, this is 2, this is 3, this is 4, this is 5. It attaches at one nitrogen, the one position. In pseudouridine, the 5th position is used for attachment of sugar. Okay, so next loop we have is this one. This loop is called an attachment loop. This is called an anticodon loop. Why this is called an anticodon? Because it contains anticodon. Anticodon, as you all know, is responsible for recognizing the codon present on mRNA, which then be capable of translating whatever the information is available on your mRNA strand into proteins. Because mRNA and protein are of different nature, that is why the word translation is used here, because we are completely translating into a complete different language. This codon consists of 3 nucleotides, as we all know. Okay, and this is bracket. Bracket means there's one nucleotide here, and which is very specific. It will always remain the same. At the 5' end, you will see that we have urine. We have our uridine. Okay, and at the 3' end, we will find any type of purine. Any other purine. Okay, it could be A is a purine, G is a purine, it could be any of them. Okay, and this is a methylated urine, I would also say. So sometimes you will find a lot of images, lot of structures saying it is ML, which is methylated purine. Okay, this is a helix, and let me tell you, this helix is very much fixed, this helix is fixed, this one and this one is variable. Now what about this? The 4th arm, these all are called arm. The 4th arm is called the acceptor arm, because as I told you, the amino acid will be accepted at the 3' end on the acceptor arm. in the acceptor arm we see a particular sequence that at the top at the three end we see amino acid which is sorry we see A, which is adenine, followed by CC. So we call it 5 dash to 3 dash CCA sequence, which is found at the 3 dash end. Okay, CCA 3 dash 5 dash. Remember that the A, that is adenine, is on the 3 dash end. Okay, and in the when we look at the 5 dash end, we don't see much interesting thing. Only thing is that the last nucleotide is going to be guanosine. Okay, last base is guanine, and what this does is this amino acid actually attaches to the sugar that is attached to our A. Okay, it has a sugar, the basic sugar that we already have. We will look into this structure shortly. Now we have just discussed about the four arms, and let's talk about the loading of our tRNA with amino acids. And this process is called charging of tRNA. For charging, first of all, what we have is amino acid. Okay, the structure of amino acid is very simple, of course, right? It has a COOH group. Okay, it then also has an amino group, and it has an R group and hydrogen. If we remove the H, we get a negative sign, which is often how it is often present, okay, in an ionic form. Then we have A. This is our amino acid, and we have ATP. What is ATP? Adenosine triphosphate. How many of people know the structure of adenosine triphosphate? The structure of adenosine triphosphate is very simple. It is a ribose sugar with adenine. And it has phosphate group. Okay, but this is one phosphate group, which is often we see when we are talking about the structure in DNA or RNA. But here, because we have triphosphate, we have more than one structure, more than one phosphate. So there are three phosphate, that is why it is called a adenosine triphosphate. Adenosine triphosphate ATP is also called the currency of the cell, because the bond between this P and P has a lot of energy, about 35 kilojoules per mole. Okay, now this negative charge that we have, the electron will attack at P, and this bond will have to be break. Okay, and the ultimate result that we get is carbon, oxygen, this is called amino acyl AMP, adenosine monophosphate, or it can also be called adenylated amino acyl. Okay, adenylated. Now this thing is only, now this is called the charging or activation of amino acid. The amino acid is now active, and this activated amino acid is quite capable of attacking the tRNA. When we see a structure of tRNA, the last at the 3 dash end, the last nucleotide was adenylic acid. Okay, so we will draw an adenylic acid. We know this is 1, this is 2, this is 3, this is 4, we got 5, we got O, P, O. Okay, and there might be another 3 dash end. Okay, and this is the last 3 dash end we got. Now we have another structure, that is our amino acyl AMP. I would suggest you all to learn to draw this structure because it is very useful. Okay, now this negative charge here attacks at the hydrogen, and if it takes the hydrogen, then it has another negative charge which then attack our carbon, which will force this oxygen to take away its electrons. This results into a very interesting structure, what we were waiting for. Now this amino acid is placed at the 3 dash end of our sugar. This is called charged tRNA, and this charged tRNA is called amino acyl tRNA.